All right, hello everybody. My name is Matt Bernstein. Today I'm going to present a recent project called the MetaSRA, which consists of normalized sample-specific metadata for the sequence read archive. And this is work that I've done with my advisor, Colin Dewey, at the University of Wisconsin-Madison. So the sequence read archive is this large repository of next-generation sequencing data. It contains raw data from over 1.5 million biological samples across over 75,000 different studies. And these data come from scientists all around the world, and they're very diverse and heterogeneous, ranging from data from cells grown in culture to tissue samples, either from a biopsy or autopsy, to cells that have undergone more involved experiments such as cell differentiation. And with this heterogeneity comes unprecedented opportunity to study various biomolecular processes and how they relate to a number of phenomena of interest to biologists, including phenotype, disease, tissue, cell type. The SRA contains a number of different data types, such as genome sequencing for studying genotype, RNA-seq for studying gene expression, ChIP-seq for studying DNA, protein-DNA interaction, among others. However, standing in the way of this opportunity is the fact that the metadata is very poorly structured. And this poor structure arises from the fact that there's no standard for which data submitters are required to adhere when they submit their data. Naturally, this leads to many problems, including synonyms. So here, for example, fibroblast of lung is the same thing as a lung fibroblast. There are misspellings. So here there should be an E between the R and the S in foreskin fibroblast. There are references to outside information. So for example, here, BJ-ATCC, CRL2522, that's the name of a cell line. A scientist will often just label their sample with the cell line name as a shorthand for all of the biology associated with that sample. But somebody from outside that domain would have to consult some kind of external knowledge base that tells you about the biology of that cell line. And there is one aspect of structure that is imposed on data submitters, and that's that submitter must encode their metadata as a set of key value pairs, where a key describes a property and the value describes the property value. So here's a set of key value pairs from a sample in the SRA. For example, the property cell type has the value fibroblasts. And here you can see a number of the issues that I described before. There's the misspelling in foreskin fibroblast. There's the reference to outside information with the name of a cell line. There's also different types of data here. So there's numeric data with 72 hours post-seeding and passage 5. Let's see. And passage 5. And there are different relationships between the keys and the values. For example, the value no is negating the key lentiviral transgenes. So we sought to normalize all of these key value pairs, all of this metadata, by encoding the metadata into a new schema inspired by the schema that's used by the ENCODE project for storing their metadata. And we broke this encoding task, or this normalization task, into 3 subtasks. First, we annotate samples with terms from biomedical ontologies. Second, we extract real value properties. And third, we label each sample with a sample type category. So I'll discuss each of these in a little bit more depth. So first, we annotate samples with terms and ontologies. And as was described in the last talk, an ontology is a structured vocabulary. So it consists of a set of terms or entities, as well as relationships between those terms and entities. So here's a very small subgraph from the Uberon ontology that describes anatomical entities. And the blue edge indicates an is-a relationship. The red edge indicates a part of relationship. So some of the knowledge encoded in this graph is that the cardiac ventricle is a cardiac chamber, which is part of the heart. And we label samples in the SRA with terms from ontologies that describe diseases, anatomical entities, cell types, miscellaneous experimental factors, and cell lines. So existing approaches that attempt to label metadata using terms from ontologies or any kind of standard vocabulary approach the task as that of named entity recognition. So named entity recognition is the name in computer science for the task of finding entities in natural language text. So here's an example key value pair. Source name, human fetal lung fibroblast, MRC5 cells, tetra BPY treated. A named entity recognition program might find the string fetal and map it to the term late embryo in the Uberon ontology, which describes anatomical entities. might find the string lung fibroblast and map it to the term fibroblast of lung in the cell ontology. It might find the string treated and map it to the term treatment in the experimental factor ontology. And we attempted to accomplish our task using one of these existing programs that takes the named entity recognition approach. But we found a number of problems that resulted in both false positives and false negatives. Now, to demonstrate some of these problems, I'll show you some examples. So here, this key value pair, disease none, a named entity recognition program might map the string disease to the term disease in the disease ontology. But as you can see, the value none is negating the key. So this is, the author is trying to say that this is not a disease sample. So this is a false positive. So a named entity recognition program won't by default take into account the relationships between keys and values and key-valued structured metadata. And the second example is the key value pairs disease breast cancer, tissue blood. Named entity recognition program might find the string breast and map it to the term breast in the Uberon ontology. We would call this a false positive as well. By labeling the sample with the term breast, that indicates that the sample was extracted from breast tissue, whereas as described here, the sample was a blood sample. And so we'd call this a false positive as well. And the third and final example, take the key value pair cell line MRC5. A named entity recognition program might map the string MRC5 to its term in the Cellosaurus. This is correct. However, there are a number of false negatives that we would also like to retrieve, namely male organism and Caucasian, due to the fact that the cell line was extracted from a male Caucasian. And by default, a named entity recognition approach won't take into account external knowledge bases that you can use to label the sample with extra terms that should map based on the information that's explicitly mentioned in the metadata. So we approach this task as an inference task where we're trying to extract terms that describe the underlying biology of the sample. And we use named entity recognition towards this end, but we're really trying to go beyond just named entity recognition. And the solution that we have come up with uses this graph data structure where different nodes in this graph represent different things. So the node on the left-hand side, this parallelogram node, represents the input, the key value pairs, and rectangular nodes on the right-hand side represent ontology terms that we output, and then a path from the input to the output represents a derivation. And we've designed a pipeline that consists of a number of heuristic procedures that build up this graph data structure. And this graph, so it maintains the provenance of each derived ontology term, and it allows us to reason about which ontology terms really describe the underlying biology. And it also allows us to reason about which extra samples we should label the sample with. So just to give you a flavor of how we might use the graph, here's one step in our pipeline. So what the pipeline does is it constructs two graphs, one emanating from the key. So in this case, the key value pair is sex F. And it searches the graph emanating from the key for the ontology term sex. And if it finds that, then it searches the graph emanating from the value. And we have a number of synonyms that we know are synonyms only in a certain context. So the letter F in this context likely stands for female, whereas if the key didn't encode for sex, F might not stand for female, and we shouldn't label the sample as female organism. And so we compared this pipeline, which we call the MetaSRA, to 3 existing programs that take the named entity recognition approach: Zuma, Sorta, and the Bioportal Annotator. And we compared all these tools across all 5 ontologies using error rate and recall, where error rate is the fraction of terms that we output that are wrong, and recall is the fraction of terms that we want to output that we do output. So the further left you are on the left graph, the better, and the further right you are on the right graph, the better. The red dot is the MetaSRA, and we really sought to minimize error rate while maintaining competitive recall. So we really wanted to restrict the number of wrong terms that we label each sample with. And you can see we maintain this low error rate while competing with the best performing methods in recall. So next I'm just going to talk briefly about the second subtask, which is extracting real value properties. And so we represent each real value property with a tuple, where this tuple stores the property, the numeric value, and the unit, where the unit comes from the units ontology. For example, the key value pair age 20 years old maps to the tuple age, 20, and year. So I'm not going to go into depth on how we do this, but we take advantage of that graph data structure that I described before, and we've achieved precision of 0.98 and a recall of 0.67. So high precision, and there's still a little bit of work to be done in boosting the recall. And lastly, I'll describe the third subtask, labeling each sample with a sample type category. So the ENCODE project, they label each of their samples according to what broad kind of sample it is. And they use 6 sample types, they being tissue, stem cells, cell line, induced pluripotent stem cells, primary cells, and in vitro differentiated cells. And the reason why you might want to label a sample with their sample type is that the ontology terms alone often lead to ambiguity as to what kind of sample it is you're looking at. So take, for example, a sample that's labeled with the terms fibroblast and stem cell. In that case, there's ambiguity as to whether the scientists started with stem cells and they differentiated them into fibroblasts, or whether they started with fibroblasts and induced them into potency, thereby creating an induced pluripotent stem cell line. And so for that reason, we found this to be an important task. And so we took the categories used by the ENCODE project, and we more rigorously defined them with this flowchart. This flowchart describes various protocols that you can perform on a biological sample and what sample type you would have after performing that protocol. So since there are relatively few of these sample type categories, this task was amenable to machine learning. And so we extract features from the key value pairs from the set of n-grams as well as from the set of ontology terms that were output by the methods I described previously. And then we trained a classifier to classify each sample type. So here's the row normalized confusion matrix, where element ij represents the fraction of samples that are of type I that were predicted to be of type J. So the higher the numbers on the diagonal, the better. And you can see that seems to be the case. And the overall accuracy is 0.84, is what we have so far. Okay, so we took all these approaches. We ran them on over 150,000 human RNA-seq samples in the SRA. And the normalized metadata is available to query and download at metasra.biostat.wisc.edu. And if you want to dig further into the methods that we use, you can read the paper in bioinformatics. So if you go to the website, there are a number of features that we've implemented to facilitate querying the SRA's data. One of these features is an autocomplete feature, where as the user is typing, we suggest ontology terms that they're likely searching for. So one challenge that we had in devising a website is that a user coming to the website doesn't necessarily know what terms exist in the ontologies that they can use to search our new database. And so this autocomplete feature helps in that regard, in that we show the ontology term that we think they're searching for, as well as terms that are ancestral, that is more general terms, as well as descendant terms, more specific terms. So this gives the user some information as to what else is in the ontology that they can also search for. And then the results we provide in a concise yet informative format, where we provide both the raw metadata, the raw key value pairs, along with the extracted ontology terms in these colorful rectangles that are colored according to the ontology that they belong to. Okay, and then lastly, I'll just describe briefly some ongoing work that we're working on. So everything that I discussed before was geared towards the metadata that pertains to the biology of the sample. And we're currently trying to normalize also the metadata that pertains to the experiment, so downstream of the biology of the sample, such as the RNA-seq protocol that was performed on RNA-seq data. And we're using the labels that we annotate, the ontology terms, to build a sample type classification algorithm, where we're taking all of this public metadata, specifically the the public data, specifically the RNA-seq data, and we're training machine learning algorithms to predict what cell type terms in the cell ontology pertain to that sample. And since the ontology is a DAG, this is a hierarchical supervised classification task, and we're using the output of the MetaSRA for training labels. So the MetaSRA has enabled this machine learning project. And so with that, I would like to thank my advisor, Colin Dewey, Professor Anhai Dwan, and Matt Ziegler, who is the lead developer who helped us build the website. And for funding, I'd like to thank the Computation in Informatics and Biology and Medicine Training Program and the Chan Zuckerberg Initiative. And with that, I'm happy to take any questions. Thanks.
Misspellings or misclassifications or something, you would have to hack those in somehow. And this framework kind of might allow you to write those hacks in. But you're probably not doing what I'm saying of coding hundreds of different hacks. What are you doing instead?
Yeah, a lot of these process steps that we've programmed in are kind of hacky. We try to make them as general as possible. So there is, for example, a stage that tries to do fuzzy string matching and mapping misspellings to ontology terms. Or for the synonym task, we have this lists of synonyms that we generate from sources or by hand. And so we try to make it configurable so the actual code generalizes, but say, for example, the synonyms would be input as sort of parameters for this pipeline. Yep.
Would it be possible to link this out to something like EDGAC or EGA, where you might have the same entities represented for the genotypes?
The same entities for their...
Meaning the same patients, participants. There are files that have the genotypes here and the RNA-seq here.
Oh, right, yeah. So I believe there are links in the SRA to these other databases, and so we provide some of those links in the website. So if that information's in the metadata, it should be possible. I don't know how.
I don't think it is in the metadata.
Oh, it isn't. Okay, well in that case, then we're not doing that currently.